Bad parasite, no cookie. Hello, and welcome to another episode of For the Love of Bugs. In our last episode, we looked at water skater beetles in the genus Sthenus, and their unusual method of escaping from predators. In order to fully understand that approach, we had to take a multidisciplinary approach in which we crossed the traditional boundaries in between physics, chemistry, and biology. Today, we're going to continue challenging those boundaries and definitions as we take a look at endosymbionts and parasites. So let's start with some definitions. What is an endosymbiont? Well, an endosymbiont is an organism that tends to live inside of other organisms, its host. And in the case of an endosymbiont, the host is generally going to be deriving a benefit from the endosymbiont that lives inside of it. A really great example is the case of Trichonympha. Trichonympha is a small, single-celled organism that lives in the guts of termites. Now, termites themselves aren't actually able to digest the wood that they eat. They lack the appropriate enzymes in order to break down cellulose, which is one of the main components of wood. Trichonympha is one of the organisms that does have the enzymes that can break down cellulose. And so by living in the gut, they're actually allowing the termite to be able to more efficiently digest the wood that it feeds on. Trichonympha, in return, gets a nice place to live that is generally free from predators. A parasite, on the other hand, is something that tends to cause damage to its host. Now, parasites can live on the outside or the inside of their host, and they tend to cause damage in one of two ways. One way is that they can cause damage by feeding directly on the host itself. An additional way is that they can cause indirect damage by feeding on the food that the host would otherwise be eating. And this can lead to things like starvation and malnutrition. A really great example of a parasite is the human tapeworm. Now, tapeworms live on the inside of the intestines of their hosts. There they feed on the food that the host would be consuming. And in large enough numbers, or at large enough sizes, tapeworms can cause things like malnutrition. But that's not the only damage that tapeworms can cause. In rare cases, tapeworms can actually migrate, so that instead of ending up in the intestine, they actually migrate over to a place like the brain, where they start to cause direct damage to their hosts as well. So that's easy, right? Parasites equal bad, endosymbionts equal good. All you need to know to be able to differentiate between a parasite and an endosymbiont is to look at the effect that it has on its host. Well, unfortunately, nature isn't always so very good at clear lines or even static lines. And a really great example of this is to look at the mitochondria. Now, mitochondria is an organelle that's found inside the cells of virtually all eukaryotic or complex cells. There, they provide a gigantic amount of the cell's energy and are frequently called the powerhouses of the cell. However, unlike a lot of other organelles inside the cell, and you can think of the nucleus here, the cell doesn't actually create the mitochondria itself. Instead, they reproduce from other mitochondria, and they even have their own DNA. And this is because at one point in time, mitochondria were actually free-living bacteria. So how does that happen? How do you go from being a free-living bacteria without a care in the world on over to being stuck inside of another cell in the form of an organelle? Well, there's lots of ideas as to how that might have happened, but some researchers have an interesting theory. They think that the mitochondria may have actually started out as a parasitic bacteria. And there's actually evidence for this. There have been multiple genetic studies that have indicated that the closest relatives to mitochondria may actually be the intercellular parasite Rickettsia. Now, Rickettsia is a bacteria that is known to cause diseases such as typhus and Rocky Mountain spotted fever. Now, this is far from being conclusive. There's other studies out there that place mitochondria DNA over with other groups, including bacteria that are not parasites. It remains an interesting concept, though, to think that perhaps the thing that makes complex life possible, that makes multicellular life possible, may have actually started out as a disease. But wait, there's more. Research is also being conducted that shows that parasites occasionally do provide some benefits to their host. Now, this benefit often comes in the form of competition between different types of parasites. In the wild, it's not uncommon for a host to be infected with multiple types of parasites. This can cause negative outcomes for the parasites themselves, because it can result in things like competition for resources, and sometimes even shorten the lifespan of their host, which basically reduces the amount of time that they have a home. A really great example of this is actually monarchs, and the competition that exists between a protozoan parasite, Ophiocystis, and a fly parasite. Now, the protozoan parasite starts out life as a spore. And you can find these spores on the leaves of milkweeds, or sometimes even on the eggs of the monarchs themselves. As the monarch caterpillars hatch out of the egg, they'll then start to eat the remains of the egg and the leaves surrounding it. And that's where they'll then digest or ingest the spores of the protozoan. Once inside the gut of the monarch caterpillar, the protozoan will then activate out of the spores and start a process of migrating from the gut of the caterpillar on over to a point that's just below the skin. There, it will feed and reproduce. Once the caterpillar then transforms on over into a chrysalis and starts developing on over into a butterfly, the protozoan will then start its next phase and start migrating on over to the scales of the newly developing butterfly, and that's where they'll form a new cyst or a new spore. So that as the butterfly hatches out, it tends to be coated in these spores. As a female butterfly is then laying eggs on the host plants, on the milkweed, these spores will then fall off onto the leaves and the eggs and start the whole life cycle all over again. The fly, on the other hand, is a parasitoid. This means that what the fly will do is it will find the monarch caterpillars, it will lay the eggs onto the monarch caterpillars. When the fly larvae hatch out of these eggs, they start burrowing into the caterpillar. And these fly larvae develop a lot faster than the monarch caterpillars do. And because of this, after a point in time, they'll have been able to consume almost the entirety of the insides of the caterpillars. At that point, they'll rupture their way on over through the skin of the caterpillar, then fall off of wherever the monarch is and start burrowing into the ground, where they'll then turn into a pupa. Now, these two very different life cycles actually put the two parasites into direct competition. In order for the protozoan to be able to go through its life cycle, it has to have the butterfly, the monarch butterfly, go through its own complete life cycle. If the monarch butterfly dies before it has a chance to reproduce, then the protozoan will die with it, and its life cycle will be broken. The fly, on the other hand, doesn't care. The fly kills off monarchs while they're still in the caterpillar stage. So this means that there's a very strong advantage for the protozoan to do something in order to try and put a stop to the fly. Because if the fly succeeds in killing off the monarch before it becomes an adult butterfly and goes through its full life cycle, then the protozoan will die along with the butterfly. And there's some indication that this may actually be what's going on. There are some preliminary studies that have indicated that the fly larvae actually experience lower rates of survival in monarch caterpillars with moderate levels of infestation of the protozoan parasite. This work is still extremely preliminary, though, and the mechanism by which the protozoan may be using to try and control or stifle the fly larva is not understood whatsoever. So please do make sure to take this all with a grain of salt, and as always, we'll include links in the description below. And it's not just monarchs in which we're seeing signs of competition between parasites. We're actually also seeing some indications that this may be going on in humans as well. A recent study has come out that has indicated that hookworms may actually be effective at controlling certain species of malaria-causing parasites. In another study, this one done in mice, it was found that roundworms may actually be effective at controlling irritable bowel disease by competing with certain types of bacteria that may cause that disease. Now, these studies are fascinating in their own right, but in order to adequately cover them will require more time than we have for this particular episode. So what we'll instead be doing is providing the links down below in the description to let you guys check them out on your own. So parasites may not actually be all that bad after all. Not that this will really change most people's opinions of them. When I started first getting interested in entomology, one of the most frequent questions that I would get was, what good are mosquitoes? Why do we have to have them? And even to this day, this still remains one of the most frequent questions that I get as an entomologist. And so it's that question that we'll be addressing in next week's episode.
Alright, that's it for today. If you enjoyed the video, please remember to like, subscribe, and share with your friends, family, nemesis, evil twins, etc. If you have questions or comments or just want to say hi, you can find links to our social media and email in the description below. And remember, alternative viewpoints are welcome, trolls are not. Thank you guys for watching, and until next time, keep your eyes on the little guys.